presentation today. We are going to do the fecal float presentation tomorrow, and we're going to do a lab tomorrow. Okay. Be sure that you are studying for your test on Friday. Okay. That test will be on Schoology. What? I spelled what? I just might if she leaves my house.
I don't want to get out my bag. These are not ugly. You got bright orange. Yellow for pee. Orange for other pee. That's smart.
Okay. Here we go. So, part one. Okay. Part one. So what is urinalysis and why is it important? Okay. Urinalysis is a routine test that reports the physical and chemical properties of urine. It diagnoses metabolic diseases such as diabetes mellitus. Okay, and it's used to assess the health of the kidneys and the urine system, basically the renal system. Okay, it helps to reveal problems in other organ systems as well, not just the renal system. Now, a urinalysis does not only have to be conducted on a sick patient. It can be conducted as routine screening on a healthy patient. Okay. Everybody understand so far? And like I said, I know this PowerPoint is on Schoology. If you want a printed version, I will print it out for you and give it to you tomorrow. Okay, you just need to let me know so I know how many copies to make. Because I don't want to waste trees or resources. Okay? Just let me know. I know there are people writing this down. How far along are we? She's still taking her test. Oh, in the morning? Yeah. Okay. Can I move on, or would you like me to wait? Uh-huh. Under med app. I just don't want the hour to be up. Yeah, the first class I finished like 5 minutes before I had to leave, and it was about an hour. Maybe it'll be faster. Maybe this will be faster, but I don't know. Where are you on here?
Just like toppling on
Okay,
each
all
other.
right.
That'll probably do it.
So, how is a urinalysis test performed? Okay, there are 4 parts that you have to remember. You have to conduct all 4. It's not just one. You have to assess the appearance. You have to look at the color and the turbidity of the urine itself. Turbidity refers to cloudiness, the urine. You need to measure the concentration or specific gravity of the urine. Okay, you need to measure pH and analyze the chemical composition. And you need to examine the cells and the solid material present in the specimen or the sample using a microscope. Now, for this last one, when you're using a microscope, it requires that the sample has been centrifuged. Okay, you have to put it through the centrifuge so that all of the denser material goes down to the bottom. Once the sedimented or settled material is at the bottom, then that is gathered and smeared on a slide and then stuck under a microscope for observation and for analyzation. Okay?
This is concentrated or cemented. Is it that depends on the tube that we put it in?
The tube that you put it in? No. So what it means by sedimented or concentrated, it means, okay, so you have a test tube, right, that has cloudy material in it. That means that that urine has particles in it that's not urine, okay? So what you're trying to do is you're trying to isolate those particles, you're trying to separate them, so that action of centrifugation, yeah, it makes all the particles that are heavier go down to the bottom of the test tube. It doesn't matter what kind of test tube it is, really. Okay?
And then you grab it and then you put it on space.
Yes and no. You will go over all of that. Okay? Promise. We're going to go over it today. All right. So let's talk about the first part, assessing colors, right? What do the changes in colors mean? Well, here I have made a chart for you. Okay? We have our colors clear to very pale yellow to light amber, dark yellow, any other color than yellow, such as orange, red, brown, or black. Okay? Let's start with the first one, clear to very pale yellow. This indicates that the patient is drinking a lot of water and urinating frequently. You may think, Miss, that sounds pretty healthy to me. No, because this, if it's super clear or very pale yellow, that means that the kidneys could be diseased, or there could be a disturbance or a disorder interfering with the ability of the kidneys to actually pass concentrated urine through, okay? Meaning that maybe the kidneys are filtering out too many particles, right? And not letting them pass through. Those particles are going to build up, right? So that's bad juju, right? You don't want them getting stuck. Now, is this normal or abnormal? It's abnormal, right? If we look at the next one, pale yellow to light amber. This one typically signifies that there's proper hydration going on and that their urination intervals are at a healthy time frame from each other, okay? What is the probable diagnosis? Nothing. They should be healthy with these two colors, right? This is normal, right? That's its category. Let's look at dark yellow. Definitely need to drink of water. Because the dark yellow signifies dehydration, right? This is an abnormal reading, okay? Any other color than yellow? Red, orange, yes.
I wanted to ask if, yes, they're dehydrated, but would there be any, like, something, would there be anything underneath that's causing them to not want to drink water?
Yeah, so plenty of factors go into a patient not drinking water or enough water. It could be that they are experiencing an infection in their body. Typically when an animal is sick, they go off of water and feed. Also, another thing, it doesn't always have to mean that they're sick. When animals travel, they go off of their feet, they're off of their routine, okay? So it's just different things. You have to think about what this animal has been through. Think of all of the possibilities and try to deduct which one would be the most likely. Okay, but it doesn't always mean that they are sick. It could just mean they're off their routine. My dog tends to get dehydrated when Daniel's out in the field because he doesn't like that Daniel isn't home. And so he refuses to drink water or eat. Like I have to force feed him, right? Because then he'll really get sick. But yeah, okay, it doesn't mean he's sick, it's just patterns are off, routine is off. Okay, so you're welcome. Any other color than yellow, orange, red, black, or brown, right? This means that the urine contains substances that should not be in the urine, okay? It could signify that there's injury or there's an underlying disease. You know, you'd be surprised how many times labs come back and they're like, well, we found substances in the urine that shouldn't be there. Let's go ahead and do further testing. and they find either cancer or they find something else that's wrong. Okay, yes?
What is red urine or something like that? Be like parvo or something like that?
Red signifies that there's blood in the system, so whatever disease or parasite causes blood cells to be shed because of either piercing or, just disintegration of the inside. That's why it would be red. Parvo is, I can't remember what it affects. I mean, if they say that parvo is indication of, or is indicated by red urine, could be that it's causing an internal bleed of some sort. Okay. This is of course an abnormal reading. Okay. Any questions or comments so far? Okay. Is this making sense? Yeah. Okay. So, what do changes in turbidity mean? We talked about color, now let's talk about turbidity. Well, what's turbid? Of a liquid, it's cloudy, opaque, or thick with suspended material. Okay? I've given you a chart of the scale, and I've also given you examples of turbidity here. Clear, typically you can put it up against a book or newspaper and read through it. Okay? Slightly cloudy, a little less so, right? It's a little harder to read. Cloudy, definitely can't see anything through there. And turbid, yeah, you can totally tell that there's stuff in there, right? So, with increased cloudiness, it indicates that there are cells or solid materials present in the urine. Okay, these can be blood, inflammatory cells, crystals, mucus, debris. I know you're probably going to backtrack, Miss, crystals, what the heck? Well, sodium crystals, calcium crystals, minerals tend to crystallize, right? Sugar crystallizes, right? So those things can be found in there, all right? Only the examination of the sediment itself, once it's been through the centrifuge, can actually tell us what is going on. It can tell us the significance of what we have found. Yes, ma'am?
If it is slightly cloudy, is that okay, or does it always have to be clear?
So, typically, good question, that brings me to my next point. Most species will be anywhere between clear and slightly cloudy, okay? It's never going to be one or the other, really. Horses and rabbits, however, are the exception. They will always appear on cloudy, okay? They'll always appear on cloudy. So,
Horses and what else?
Rabbits.
So if it's clear, it's like weird.
Oh yeah, definitely something's wrong. Okay. Any other questions? No. Are you sure? Okay, so what does a change in color mean? Anybody give me a category? Yes. Okay, red means there can be some sort of internal bleeding. Anybody else? It's okay, brain farts happen. Let's go with Zoe.
Dark yellow means they're dehydrated.
Dark yellow means dehydration, okay. You wanna try again here? Okay, one more minute. Yes.
Well, because I don't know.
Go ahead, Simone.
Light yellow means a lot of like failure in the kidneys
How so?
because they're not filtering they're not filtering the substances. Like they're
Okay,
getting, like,
not
clogged.
necessarily not filtering. They are filtering too, not like too well, but they're just not allowing those sediments to pass through,
Yeah.
which need to come out, right?
That pale yellow or like slightly darker than that, it means they're not because of periods.
So, pale yellow to light amber, yeah. And then it's clear to very pale yellow, right? Yeah, I know. It's weird with the colors. I hate to be colorblind and have to do that. Okie dokie. So believe it or not, my husband is colorblind, and so he has trouble in the lab sometimes. He's learned to identify things through using other things, especially when he's looking through microscopes. But okay. All right. So let's talk about specific gravity. Well, what is it? How many of you have ever heard of specific gravity before? Okay. Well, specific gravity can also be called urine specific gravity, which leads me to its acronym, USG. Okay. It measures the solute concentration of the urine, which indicates the kidney's ability to concentrate or dilute the urine. Okay. So it tells us how much sediment is in the urine. Okay. This obviously tells us if the kidney is working properly. If it's too dark, if it's too concentrated, something's wrong. If it's too clear, too diluted, something's wrong, right? It needs to be within that sweet spot. Now, how is it measured? We use a refractometer, okay? We use a refractometer, and it's calibrated. People will tell you, oh, it's calibrated per species. No, it's not. It's calibrated per solution, okay, or by solution. By distilled water, because distilled water has been filtered and filtered and filtered, okay, and there aren't any particles left in it, okay, so it'll give us a true value of 0. There's a glass window here, and then there's this plastic cover. It's like a telescope or a monocular, and you look up through it, and you can see a scale in there, okay. That scale will look something like this. There are different refractometers with different measurements. This one is salinity. We're not using that one. Ours is going to say urine specific gravity, or urine SPG, or USG, okay? That's what ours are going to say. Okay, so I went over, you hold it up to a light source, and you look at it through the eyepiece. And the USG is recorded at the light to dark interface. So where the dark portion meets the light portion, there's that line, right? That's the measurement you're going to read. And you have to pay attention to which scale you're using, because there are going to be like 2 to 3 different measurements or scales in there that you're going to have to pay attention to. Remember, you're reading the one that has urine USG or urine SPG, okay? Yes?
So how exactly is that measured the concentration? Like, how do you...
It helps us determine, I guess, a percentage, a ratio of how turbid or cloudy the urine actually is, okay? So sometimes when you're looking at it, the color, you're looking at appearance, can't really tell. The next step you're going to want to do is use the refractometer, okay? Because there's scales, there is a range. If it falls below a certain number, something's wrong. If it falls within the sweet spot, we're okay. If it's something other than that, then you know something's wrong. So you're looking for that sweet spot. And on the next slide, I have another chart for you. Okay? Yes?
Where do you put it in from?
Okay. May I? And I have a specific slide on how to use this. So I'm going to go over it really quick just because that question was asked. So what you do is you lift up this plastic cover. You're going to take a pipette, dip it in your urine, suck up some urine. You're going to place 3 to 4 drops. You can do 2 to 3 also. This one doesn't have a little circle, but generally speaking, some refractometers have a little circle where you put it. It's like a little divot. You're just going to put it in there. But if not, you're just going to put it on the glass panel, this blue panel. You're going to close your cover, and it's going to spread out the entire sample of urine that you have on there. And all you're going to do is look up, and you'll see the scale in there. So I don't know if you've looked through it. Okay. So if you want to pass it around.
Yeah.
Yes.
Why is it slanted?
For the light. Yes.
When you're on the
They use it for human use as well.
Would it be better if you literally pee on a cup and you just put the drops yourself? Like just pour the glitter? Or you literally pee on it?
Oh, you don't pee on it. You use a pipette. So a little a little dropper.
Yes.
Okay.
This is not used to
No, that'd be a mess.
Don't
That'd be a mess.
like 2 seconds.
Okay.
Was not pilot.
Okay. So why is specific gravity important? Well, it tells us if the job that the kidneys are supposed to do is in line, is functioning. The kidneys are supposed to maintain the body's water level within a narrow limit, within the sweet spot, right? So anything, it's a very fine line what the kidneys do. So if anything falls outside of this area, then you can bet that something is wrong. Okay? If there's an excess of water present, what if, right? The kidneys, that means that the kidneys are allowing excess water to pass out in the urine, which is diluting the urine too much. It's letting too much water escape. Your body needs water to function, right? At this point, it's letting too much water pass through. If a shortage of water is detected, such as dehydration, the kidneys will reduce the amount of water being passed through, right? So in essence, that is also not allowing for the proper concentration to pass through as well. If water's being withheld, you bet that the other sediments are being withheld, right?
The sediments or the urine concentration itself, right?
Yeah. Kidney stones, right? It's a buildup of crystallization and sediment, right?
Wait, should I have a question?
Sure.
So you know how humans like sometimes have like a little bag here, and someone's like, oh, it's like my kidney stone bag or something. How does that work?
It's not
I'm so
necessarily a
confused.
kidney stone. That is a colposcopy bag, and that is for their intestinal tract. It helps them with secreting the excrement, right? They have to, because it doesn't go out anywhere else. They could have a hole in their intestinal lining that they have to seal off the intestines, and now instead
Where's
of
this
pooping
Carolina
out the
kids
blood,
to the right?
they excrement comes out through that bag.
Miss
I remember where I heard it. There was like some ants talking about like, oh yeah, my kidney stone bag or something.
It could be that the kidney stone, when they were passing it, caused a rupture of some sort, caused damage, and now that they have to have that colposcopy back.
Oh.
Yes.
Kidney stones can be suffocated or peed out, right?
Only peed out.
They can't remove it.
They don't, honestly, it has to be like a super, super dire situation, but normally it's just passing through.
Wait, I have another question.
Yeah.
Isn't it hurt like super bad to
So,
get kidney stones?
wasn't soda do that too? Like drinking a lot of soda
Yeah.
causes kidney stones?
Mhm. Because of the amount of sugar, the sugar crystallizes, right? Yes.
Don't they say like if you hold in your keys and there's that also?
It's bad for your urinary tract, and you have greater risk of infection, right? Yeah. Any other questions? No? Jonah? Okay. So, we have our interpretations, right? We have our reading. If it's less than 1.008, something's wrong. It's, bear with me. Hypocynuria. Yeah. I can't pronounce it, whether I try to do it slowly or not. If it's between 1.008 and 1.012, it's isothynuria. Typically that's a little more within the normal range, right? It's only a 4-point difference there.
That's using the scale.
Mm-hmm. And if it's greater than 1.012, it's is in the hyper stage. So you're always looking for the sweet spot. You're always looking for it to be in the middle. Okay? So let's move to how to use a refractometer. Okay? Like I said, you're going to place several drops of the sample liquid on the angled prism. Okay? You seal the clear plate on top of it, and you look through the eyepiece while pointing the refractometer to a direct light source. Okay? And you follow the line of refraction across the scale to determine the specific gravity in the patient. Now, when the refractometer, as many of you have already seen, when it's empty, it's going to be all blue. Okay? It's going to be all blue. When you calibrate it, meaning when you put distilled water in it to be sure everything is okay, right? It should be at that 0 line. It should set right at the 0 line. Okay? Just 1 drop of water. Okay? It should set at that 0 line. Once it's at that 0 line, then you're going to add in your urine, or the urine, not yours, but the urine, okay? And it'll raise that level, okay? And it'll give you a reading, and that's where you have to make the best educated guess, because those hash marks are really hard to focus on sometimes, okay?
Do they ever move?
The scale, do they ever move?
Not the scale, but the urine within it. It doesn't.
No, it shouldn't. It shouldn't. It's not beaded. Yes, ma'am.
I'm so confused because so you put the urine here, but how does that have to do with like the little measuring needle here? Like I get like yes, no, I'm so confused.
Okay, articulate your question better so I can better answer.
So the urine's right here, right?
Correct.
And then you got the little image like right here.
Mm-hmm.
So how is it possible to like see the measurements when this is slanted like this?
You're okay, look through it. Point it up to a light. Okay, do you see numbers?
Barely see it.
Okay, you can adjust it. You can adjust the scope.
Okay.
You can like move the eyepiece like you would binoculars.
Oh. Yeah.
Okay, so it comes into focus. All right. So when you put your urine sample on there, once it's been calibrated, you're going to first put your distilled water. Once it brings that up to 0, then that's when you can start your measurement. You put the drops of urine on there, and it will raise that refraction of light, okay, to whatever level it's going to be at. Mm-hmm. Yeah, and we'll practice with the refractometer, which is why we need urine. Okay? Which is why we need urine. That way you can see in real time how it works. Okay? To give you some practice on how to read it. Okay? Is the concept understandable though?
Yeah.
You sure? I know, I think you have to see it done in order to explain it.
Okay.
And there's no way for me to do that right now. Yeah. Okay.
Ice cream.
So if you take a look at this sample or example, if you have your Schoology up, your PowerPoint, take a look at that picture that's in the black circle. Okay. We have our refracted light line right here, right? It's right in line with this ND serum, count serum platelets at 1.35. This is not something we're going to be paying attention to, but this is a guide I'm using, okay? You're going to look at the scale that's labeled urine SPG, which is on the far right-hand side, okay? And if you look at the scale, everything is in increments of 10, right? 1.0, 1.01, 1.02. So we have 1, 2, 3, 4 hash marks there, which typically indicate that there are 5 spaces there, meaning that each space is valued at a 2. Okay? So if you look at the picture or picture here, we're right above the 1.03. What is the value of this particular sample here? Can anybody tell me? Does anybody want to come up and take a closer look if you're having problems seeing?
You said they're at the values of 2?
Mm-hmm. Each space has a value of 2. .036. Yeah, exactly right. It's 2, 4, 6. The line is at that 6 mark, right? So the reading would be 1.036. Looking at our chart from the slide beforehand, where does 1.036 fall?
The first one.
Greater than hyper. It would be hyper.
So
Okay.
30.
The terms in that interpretation portion, you all are going to have to define. Okay, just so you know. All right. So, I have a video. I think I have a video. Hold on.
Wait, look.
Yes. I do. Thank you.
Earring. One earring. Because I rolled out right there.
Oh, I think I closed it out.
Small
Yeah, I closed it out. Sorry. I will find it again though. Let's continue on. Okay. So, what is a chemical? Ooh, wait. Like a dummy. No.
I was gonna say, isn't that linked to us?
Like a dummy.
Hi, I'm Dr. Evan Matthews. I'm here at Montclair State University in Exercise Science Lab to show you how to do urine-specific gravity using a refractometer. Right, all you have to do is take the refractometer, you're going to open up the top, and you're going to put 2 to 3 drops of urine using a transfer pipette right in there's a little sort of circle hole in underneath the glass of the refractometer. Alright, so make sure you get it onto that hole, and then you're going to close the lid back over, and you're going to see that the urine is kind of spread out across that piece of glass that's off the refractometer. Once that happens, you can then take the refractometer, hold it up to your eye like imagine sort of like a telescope, and just walk underneath of it as some sort of bright overhead light.
Thank you.
Refractometer. State University in Exercise Science Lab to show you how to do urine-specific gravity using a refractometer. Alright, all you have to do is take the refractometer, you're going to open up the top, and you're going to put 2 to 3 drops of urine using a transfer pipette right in there's a little like sort of circle hole in underneath the glass of the refractometer. Alright, so make sure you get it onto that hole, and then you're going to close the lid back over, and you're going to see that the urine is kind of spread out across that piece of glass that's off the refractometer. Once that happens, you can then take the refractometer, hold it up to your eye like imagine sort of like a telescope, and just walk underneath of it as some sort of bright overhead light. Alright, so make sure that you are under a light because it's going to use that light to shine through the liquid, and so it's easy for you to see what the specific gravity is. Alright, so again, walk under the light, look into the refractometer like it were a telescope, and then you're going to look for some different scales on the refractometer. On this particular refractometer, the specific gravity scale is on the lower left-hand side. So all you're going to do is look at the lower left scale where the specific gravity is, and look for where the black line goes across the scale and read off the scale. And that is the specific gravity of the urine. Alright, so if for some reason there's not a very distinctive black bar, I would just sort of open up the refractometer, clean it off, and then start over again. It usually means maybe you got a little air bubble or didn't completely cover the glass with the urine, something like that. But if you just start over again, it will typically work the next time around. All right, so once you finish the measurement and you've written down your value, you can then open the top of the refractometer, take some sort of tissue like a pin wipe, something that's not going to leave any dust particles behind because you don't want to affect the next measurement, and soak up all the urine that you put on the device. Once you've done that, you can clean off the device with an alcohol swab and simply wait until the alcohol is completely evaporated before storing the device or trying to use it again. All right, so that's all it takes to clean the device and get it ready for the next time. Be sure to also clean off the countertop underneath wherever you were working in case any urine got spilled. So urine-specific gravity is really good for assessing hydration status. Typically, any value above 1.020 is considered dehydrated. All right, so you can see here that I ran 3 different urine samples, 2 of which were dehydrated, 1 of which was well hydrated. The sodium levels within these samples matched up with the specific gravity measurements. So the dehydrated samples had higher sodium levels. The normal hydration or even hyperhydration sample had lower sodium levels. All right, so sodium and other electrolytes in the urine is going to be a lot of what makes up the solids within the urine affecting its specific gravity. All right, so that was a real fast overview on how to do urine-specific gravity using a refractometer. If you have any questions or comments, you can put those in the comment section below, and I'll try to get back to you. Otherwise, please come back and watch another video. Thanks.
And we will have practice with the refractometers tomorrow, okay? All right. Okay, so let's talk about the chemical analysis portion of the urine, right? Yes, like when they measure pools. Yes. The biochemical evaluation of how acidic or alkaline a solution is, in this case it's urine. So we're looking for the pH levels. Now, keep in mind that pH can change with diet, okay? Whatever the patient has consumed can change those levels, okay? And, but the thing is that with that, we have to also remember that this can also indicate an infection or a metabolic disease in the patient, okay? Cats and dogs range from mildly acidic to mildly alkaline. And I'm pretty sure you're like, well, what the heck is that? Do we have a number value? Yes, we do. Numerical value. Normal pH range should be anywhere from 6.0 to 7.5 for dogs and cats, okay? It's very important to remember. Any extremes below 6 or above 7.5 is abnormal and needs to be further tested, okay? So how is the chemical analysis performed? Well, we use a dipstick. I believe last week I handed out, or I let you all see the dipstick, right? And you got to see the bottle and it had the different colors on it. You're going to place that dipstick in the urine sample. Now here we have a picture of a cup, right? Let's say that's all the urine we have, and we have other tests we need to run. So we're not going to want to dip the stick in that particular cup. We're going to want to remove some of the urine, put it in a test tube, and then dip the stick in the test tube, okay? So try not to contaminate the sample that we have, or from further contamination, okay? Or a pipette. Typically, pipettes are in a sterile environment, so you just, or clean environment, right? Okay. So on this dipstick, each pad that's on there measures a different chemical compound or component and changes color to indicate how much of that component or that substance is in the urine. Okay. I also have a video for this. It's not this.
This this video is going to demonstrate the urine dipstick examination. The supplies that you're going to need for this lab are the urine dipstick bottle that contains urine dipsticks, or the color wheel for each of the dipstick pads, as well as 5 mLs of urine, and a stopwatch, which I'm going to be using my cell phone for this. So if you see on this dipstick color wheel here, each of the pads corresponds to a different chemical property within the dipstick. So we have glucose, bilirubin, ketones, specific gravity, blood, pH, etc. And then next to them, it has the number of seconds that it will take, at which point you'll need to read those dipstick pads. It's very important not to read them too early or too late, because it will affect the color of the color change, because as the urine sits on the dipstick pad, the enzymatic reaction continues on. So it's very important that you read them at exactly the time that they say. You'll also be reporting these on a piece of paper, which is not shown here. This is only a demonstration of how to use the dipstick pad. So you'll take your dipstick pad, and you'll see that the bottom says Siemens on it. That's the bottom of the pad, which will correspond to this white negative pad here, and going up and so forth. So step 1 is to take your dipstick, and either using a plastic pipette or simply gently dipping the dipstick pad into the urine, you're going to coat all of the pads evenly with the urine. You'll gently tap off excess urine onto a paper towel or into the sink.
Okay, really
You'll
quick.
also start your stopwatch. So that
She's using her phone here. I do not recommend using this. Tomorrow, you will not have your phones out, okay? She's touching biochemical, right? She's touching biological waste with her gloves, and she's using her phone, which is then probably going to go up to her face at some point in time, right? Please, 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 remember, that's why we have our watches, right? You're going to look at your second hand, and you're just going to time, right? That's all you have to do.
Right there.
What's up?
She doesn't watch right there.
I know, and she. Anyway, I'm pretty sure for the purpose of the video, she wants you to see the time passing, but at the same time, proper laboratory etiquette. Yes.
Are we using actual people?
Yes. Yes. That's why tomorrow I want you all to come in properly, at least hair pulled back. I don't want big jewelry hanging down. Yes. Who's pee? Mine. No. Unless you want to volunteer. No, it'll be animals, and some of you have volunteered to take a pee cup home to collect the sample, right? So, yes, that'll be that. We'll get them in a minute. Okay. Yes.
Are we gonna be able to confirm it was an hour?
You know what? I'm not going to be able to confirm that. That's why we're just going to wear. We're going to be safe. We're going to wear proper PPE. We're going to wear gloves, right? It's going to stink tomorrow too, because we're going to have poop. We're going to have fecal float. So, yes. Okay. So you can bring your Vicks Vapor Rub, got the rule, right? Or if you have peppermint oil, you can use peppermint oil on a mask. Don't put it directly on your skin, right? But you can do that. I think I may have a vial of peppermint oil at the house, so I'll bring that with me tomorrow. Okay?
I have an essential oil.
There you go. That's all you need. That's all you need. Okay. Let's continue watching this bad video.
so that we know at what time to start reading the pads. The first pad, the glucose pad, which is this brown colored one at the bottom, is the first pad that we're going to read as soon as it hits 30 seconds.
What about the extra seconds it took for her to?
So we can see that it's about to hit 30 seconds. So we'll line
Another reason you don't want to use your phone.
the negative pad up at the bottom, and we'll follow this color pad over to where the glucose color is changed. And for this, it says that it's 2,000 or more. We'll then move on to our bilirubin, and you can see that there's no color change, which is probably, this is negative. The ketone is trace. Specific gravity is 10.5. Our blood is 0. pH is kind of an orange color, so it looks like it's somewhere between 5 and 6. So I'll probably go to 6. We can see that the proteins are negative. The urobilinogen is negative.
Yes, sorry.
The nitrate is negative. And then last but not least, the leukoesterase is negative. So you'll just go through and using this as a guide,
I'll walk them.
you'll start reading your dipstick pad.
I have to go down there anyway today. Okie dokie. Let's try to continue. Let's try to finish this so that we can jump into part 2 tomorrow. Okay? Yes. 30 seconds. Yes. Yes. You need to follow the timestamps that are on there, because if you read it beforehand, it's not going to be a true value. If you read it too long afterward, then chemical decomposition has taken place. So it's not an accurate reading. Somebody else have their hand up? Yes. Go. Okay. So, these are the common constituents that are measured in veterinary medicine: pH, glucose, ketones, bilirubin, protein, heme, such as blood, hemoglobin, myoglobin, leukocyte esterase, nitrates, urobilinogen, and USG, or specific gravity. Now, these last ones here, on this last bullet, these are typically unreliable, so don't necessarily rely on this test, especially when you're trying to find specific gravity. Okay? Look at the other components. All right? That's why we have other tests for those. Yes? Uh-huh. Yeah.
So,
What's your question?
my question is, because you're handling the urine, it's like I guess the sanitary.
I mean, yeah, I understand what you're saying. You're not going to put your pencil in your mouth, are you?
No.
Okay. I mean, obviously, if you're not going to be splashing it about either, right? You're going to be as careful as you can. So, unfortunately, yeah, you're going to have to write down your test results, because you're not going to take off your glove. I mean, you could stay ungloved with one hand if it's your dominant hand, but are you going to have enough control with your non-dominant hand to perform the test? Does that make sense? It's all about comfort level. It's really how you perform, so keep that in mind. Okay? Great question. All right, so let's talk about the last component. Why examine the urine sediment? Well, it's the material that sediments or settles at the bottom of the tube when the sample is centrifuged. Okay, what can be seen in the sediment? Anything like red blood cells, white blood cells, bacteria, crystals, and tissue can be seen. Okay, red blood cells in small numbers, typically it's not a bad thing, right? It could be due to the method in which the urine was collected. Okay, if it's in large numbers, then yeah, go ahead and start worrying, right? It usually indicates major bleeding. If you see white blood cells, what are white blood cells? Anybody remember? The soldiers that do what? They fight against infection. Well, if you see white blood cells in small numbers, it's not usually a significant worry point. Yes. Yes, ma'am. Large numbers indicate inflammation somewhere in the urinary system. Okay? Bacteria. If you see bacteria in there, it's normally accompanied by white blood cells, right? Those go hand in hand. And usually, if you find bacteria also, you need to send it out to get cultured. You need to grow that bacteria so you figure out what it is and what antibiotic to use to treat it. Yes, ma'am?
My girl needed to do me like grab the cotton swab and then like grab the platelets
In a petri dish. Yes.
Is there specific questions in that?
Sometimes you can do it in house. It takes a matter of minutes. Sometimes. I know that they've really gotten better. They've really gotten better at that.
Wait, so when you put it in a little like in the thing where it like grows
The incubator.
the incubator, how long does it usually take to like grow the bacteria?
It can take maybe 30 minutes.
What?
Yeah.
That's it?
Well, because they're introducing it to a controlled environment that, it's heated, humid, right? Bacteria tends to flourish in that type of environment, so it's rapid growth. Okay. All right. If you see crystals or tissue cells, these are unique, and they can help pinpoint specific diagnosis in what is wrong with the patient, whether the renal system or anything else. Okay.
How does that do the crystal cell?
What's up?
How does that do the crystal cell?
I'll find some photos for you. Okay. So really quick, how to collect samples. Okay. There are 3 methods. Well, technically there are 4, but the 4th one I decided to log off of it. Cytocinesis. You take a sterile needle and syringe and you poke through the abdomen into the bladder. Okay. This will actually cause bleeding, right? You're not just getting the urine, you're also getting blood because you caused the piercing, right? Now, the advantage to cytocinesis is that the urine isn't contaminated. It's coming directly from the bladder into our sterile syringe, right? Yes, ma'am?
Isn't there this other thing you can also do? It's not necessarily like a needle. It's like this like very, very, very tiny tube, like this like tiny plastic tube. You just like shove it through like the penis and then you like press the.
That's catheterization. That's all.
Oh.
Okay, a sterile catheter can be passed up through the lower urinary passage, right? Through the urethra or through the pineal gland, okay? And into the bladder directly. The advantage here, it's less invasive than cytocinesis. You're not causing a puncture. However, you can still get blood cells because you're scraping that wall with the tube, causing an abrasion, okay? It does. Catheterization hurts like heck if you have somebody who's inexperienced doing it. Okay? Maddie's face. And then there's midstream flow. The process you will be using to collect your animal's urine tonight. Once you collect it, your parents are probably not going to let you do this. Put it in a Ziploc bag and try to put it in the fridge. Okay? Double bag it.
You just gotta put it in the
The poop, yes, also goes to be refrigerated.
Maybe in the freezer. No, the freezer will get frozen.
Yes.
Put it in the back. Put it in the back.
And you can do so.
Just like
Move all the
You can do so in like a glove, like pick it up with a glove.
Okay, that's actually a double.
And then, yeah, and then this is a block or 2 or 3. Yeah. It has to be fresh
Oh, I know what I
laid.
can do.
But don't let it
That's
touch the
what's
ground.
funny. There's this other dog.
Okay.
So I really need to like get her and get
Exactly. Okay, so midstream flow, right? This means that the urine is being excreted from the system. It's being expelled voluntarily by the patient, okay? And it's collected in a sterile container, okay? It's also called free catch. The advantage here, zero invasiveness, right? But the drawback is that these samples tend to have more contamination because the environment around us, especially if you're outside trying to catch the bee and it's windy, you're going to catch everything else in there too, okay? So there are pros and cons to every method, right? You just kind of have to do what you can. Most owners, most clients will bring in a sample already in a cup. Unfortunately, sometimes those are so contaminated because they had to either rinse out the cup that they're using. You know, they're like, oh, this is good enough, I'm just going to put pee in it. Or they have detergent in there because they also didn't rinse it out. Maybe they took it out of the dishwasher and their dishwasher is all, you know, wonky and not rinsing properly. I don't know. Those are the risks that we run when we get a sample from home. Okay? On this last slide, you have vocabulary terms you need to know. Most of these are defined within the PowerPoint presentation. The only three that really aren't on there are these first three here, and dehydration is not on there. You can kind of do that. Calibration or calibrate is not on there. Concentration is not on there. Alkaline and acidic are not on there. Okay, so